Welcome to the lecture on protein synthesis. We're going to look at how we create proteins in our cells. We're going to go from what we call transcription to translation and the production of a functional protein. So first off, let's review a little bit about DNA and RNA. So DNA is our double helix complex, contains our genetic code. RNA is seen in a variety of ways, variety of structures. There's three types of RNA we're going to look at in this chapter. There's tRNA. The T refers to transfer RNA. There's rRNA, and this is the RNA that makes up ribosomes. So it's ribosomal RNA. And then there is mRNA, and that's the one we're most often used to hearing if you've taken any biology class before, and that's called messenger RNA. So those are the three types we're going to look at. Key things we need to remember with RNA is single strand, okay? So it's a single strand. It contains, instead of T, it contains U or uracil. So instead of thymine, it contains uracil. So in RNA, A bonds with U and G bonds with C. While in DNA, it's A and T and G and C. Okay, and so that's important, that's important when we start going from DNA to RNA, because if DNA is an A, RNA is going to be a U, and that'll make more sense in a minute. Okay, so make sure you remember RNA is uracil. Okay? Alright, let's go ahead and move on to the next segment here. More specifically, let's look at the RNA types a little bit, what each one does. tRNA, what it does is, and we'll kind of draw it like this. tRNA, and the reason I've drawn it like this, kind of looks like a T. It transfers the amino acids in to build the protein. So if this is our amino acid, it brings those in to build the protein. Okay, so when we're coding for the protein to build, tRNA is the one that actually brings them in. It transfers the amino acid, picks up the appropriate amino acid, and transfers it in based on the mRNA to make the protein. Now, where does it take the amino acid? Well, it takes it to the ribosome, which is the, I'll use a different color here, which is the ribosomal RNA. So this is what makes up our ribosome. And we'll see we'll look at a ribosome a little more up close here in a minute. But a ribosome is actually made up of two parts. Looks like that. Okay, it has a small unit and a large unit. Now, when tRNA comes in, which we're going to go in depth, comes in to bring the amino acid, it actually binds to the mRNA, which sits in here when we're making a protein. It sits in the ribosome like that. And mRNA is messenger RNA, and so it carries the message. Okay, it's messenger RNA. It carries the message from DNA of what protein we're going to make and how to make it. And what tRNA does is it comes in and actually binds with the mRNA. It actually binds with the mRNA, and it does that through this part right here, which is called the anticodon. Now, if we think back to chapter 13, a codon is a set of three bases. So if AUG was the first one here, which you'll get used to hearing, the anticodon on the tRNA actually binds with AUG. So the anticodon would be UAC, right? It's all complementary base pairing. C binds with G, A binds with U, and U binds with A. And so that's how those RNAs function in protein production. This will all make a little more sense here in a little bit, but this gives you at least an idea of the different types of RNA and what they're actually doing in transcription translation. Alright, let's look at transcription. Transcription is going from DNA to RNA. Okay, this is transcription. The process of being able to transcribe from DNA to RNA. We're taking the coded message of RNA and putting it, or of DNA, and put it into the coded message of RNA. Because DNA can't leave the nucleus, right? No on nucleus. Yes on nucleus. The RNA can leave the nucleus. So we put it into RNA and send the message out to the the rest of the cell in order to make the protein. Now transcription occurs in the nucleus. Okay? Transcription occurs in the nucleus. Again, because DNA cannot leave the nucleus. Now to look at this a different way, this is the beginning process of going from our genotype, right, our genetic makeup, our DNA, to our phenotype, which we're not there yet, but that would be the protein. The protein is the actual phenotype, the physical expression of the gene. Okay, so we're seeing how we got, when we looked at chapter 11 and 12, we looked at all these different traits and disorders, hanging earlobes, hair color, eye color, blood disorders, whatever it may be. Those are all created by this process. So we go from genotype to phenotype. Alright, so let's look at the steps of transcription. So this right here, I'm going to draw a box around, is transcription. Okay, the process of creating that mRNA, which is the yellow one here, from DNA. Now we're going to look more closely at the actual process here on this next picture. Okay, so there's 3 steps I'm going to give you of transcription, and they're really easy to remember. The first one is initiation. Okay, initiation. So what happens in initiation is the enzyme RNA polymerase attaches to the DNA. Okay, so RNA polymerase attaches to the DNA. Now where does it attach? RNA polymerase attaches according to what we call the promoter site. Now the promoter site is a site where there is a sequence of nucleotides, so it's a sequence of DNA. So let's say the promoter site is right here. Okay, that's the promoter site where that green is. Proteins are going to bind there, and that's going to tell the RNA polymerase enzyme, so this is our RNA polymerase, to bind right in front of it and start coding. So the promoter site isn't a coding sequence. We aren't going to use that to make a protein. It's a sequence that controls transcription. It's a factor that controls transcription. Okay, so this protein actually aligns RNA polymerase on where to actually code the DNA, where to transcribe the DNA. So the RNA polymerase in initiation attaches there, and it decides which strand to describe. So once it attaches, it's told which strand to transcribe. Sorry. So is it going to transcribe this side, or is it going to transcribe the other side? Because we're going to only transcribe one side, and that's what we call the coding strand or the template strand. Okay, the other one's going to be your complementary strand or your non-template strand. So there's proteins here that tell the RNA polymerase to bind there, and then it'll begin transcription. Okay, that is initiation, and that begins the synthesis of RNA. The second step, if I clear this ink, is RNA elongation. It's really simple. We're just building our RNA molecule. RNA elongation. Building our RNA molecule. And what happens is, as our lovely RNA polymerase goes along, you can see down here as it goes along, these amino acids pop down in there. I mean, not amino acids, but bases. So let me zoom in for you here and show you what I'm talking about. So if I go on here and I go in closer and move it over right here, we can see, let me clear the ink here, we can see, if I use black here, you can see these little nitrogen bases, right, the parts of the nucleotides, those little nucleotides, and you can see them here. They all come in and bond according to their base pairs, right? G bonds with C, A bonds with T. Now, in RNA you have U, right, instead of thymine. So if there's an A, U comes in for RNA. So all these nucleotides, RNA nucleotides, come in and bond, and RNA polymerase comes along and attaches them together. As those bases come in, RNA polymerase attaches them together in order to enlarge our RNA molecule. Okay, so that's step 2, RNA elongation. I'm going to clear the ink on this page. I'm going to move this up a little bit so we can see here. Now, as we lengthen our RNA molecule, the DNA behind it twists back up. As you can see here, it twists back up. So it's not going to stay open very long. It's only going to let the RNA polymerase come along and code, and as it goes along behind the RNA polymerase, the DNA winds back up so that nothing's damaged, and we're only coding the part that we want to code. Now the last step is step 3, which is termination. So we can all tell from that name, right, it has nothing to do with Arnold Schwarzenegger and the Terminator, but it has everything to do with stopping. So what happens is RNA polymerase, so here's our RNA polymerase molecule, it reaches a spot where there's a sequence of nucleotides, so a sequence of DNA called the terminator site. Terminator, again, it's not Arnold, but it does have to do with stopping. When it reaches the terminator site, it is going to tell the RNA polymerase to stop and to detach. Okay, so terminator site is a sequence of DNA that says, hey, this strand is done, you've coded the protein, leave. So the RNA polymerase will detach, and we will have made our mRNA molecule. That is our messenger RNA molecule. Now we go through the same type of functions to create our tRNA and rRNA, but this is specifically transcription for making our messenger RNA here. Now, what we don't see is the fact that this RNA molecule actually isn't quite ready to become mRNA. At this point, we're still just going to call it RNA. Okay, it's not quite ready to be our messenger RNA. It has to go through a process between transcription and translation, which we'll look at next. So that's the steps of transcription. If we go back here, this is a more larger picture, kind of drawn out picture. If I clear the ink, remember up here is transcription, and there's those 3 steps, okay, 1, 2, and 3. Alright, remember those. Initiation, RNA elongation, and termination, and know what happens in each, okay, and know what we're producing. We're producing an RNA molecule. Now before that RNA molecule can continue out the nucleus, it has to go through what we call splicing. RNA splicing, which we're going to look at next. Here's also another picture of transcription as well. Okay, you can see how we code from DNA, from the coding strand of DNA or the template strand of DNA to our RNA. Now we're going to talk about RNA splicing. Now RNA splicing is what allows us to have a variety of proteins or traits with limited genes. For example, we have about 20 to 25,000 genes, but we have at least 80,000 proteins. Okay, so 20 to 25,000 genes code for at least 80,000 proteins. So what does that mean? That means we have to have a way of altering these genes to create a variety of different proteins. One gene has to be able to code for more than one protein. And we do that through splicing. So if I clear this ink, what happens is, let's say this is our RNA. And what's going to happen is, in splicing, is we're going to take out segments of it. And then we're going to take these segments here, so if that's 1, 2, and 3, we're going to rearrange them. And it may be 1, 2, 3, or 3, 2, 1, or 2, 3, 1. We're going to rearrange them, and that will ultimately become our mRNA molecule. Okay, and that's RNA splicing. Now I'm going to show you, or we also hear it called alternative splicing. I'm going to show you a better picture of that. So here we can see how one RNA molecule can become 2 different mRNA molecules and create 2 different proteins. And now if we look down here more specifically, we can see how we removed segments of it and kept some sections. And then we can put those sections back together however we want. So for example, if that's 1, 2, and 3, we can put it together as 3, 2, 1, 3, 1, 2, however we want to put it back together to create the protein we need to create. That's RNA splicing. Now, what's the names of these items we're removing? The ones that we're removing are called introns. So think of like intervening, introns, intervening, they get in the way. When you intervene, somebody's getting in the way. We're trying to get rid of the interveners. We're trying to get rid of those things in the way. So the introns we're removing. The yellow ones here, the ones we're keeping, are the exons. Think of exons as expressed. They're the ones that are expressed, so we're keeping them. Okay, so we're going to keep exons, get rid of introns, or splice out introns. Okay, and that allows us to have a fully functioning, ready to go mRNA, ready to go through translation. Okay, so you have RNA at the end of transcription, you go through splicing, and you get a ready to use mRNA that'll head outside of the nucleus. Now, one other thing that affects transcription is what we call transcription factors. Now, we talked a little bit about this with the promoter site, which is right here. So these two are the promoter sites. This is the proteins and the site that tells the RNA polymerase where to bind. But there's also what we call enhancers. Enhancers are proteins that help line up everything. They basically control the efficiency of the gene. Okay. So for example, here we're looking at two different organisms. This is a different one here, and this is different. That's organism 1, organism 2. Two different organisms. They have the same exact gene that codes for the spine, but because of the difference in their enhancers, one of them produces less vertebrae than the other. And that's all due to the transcription factors called enhancers. So see how on this one it's lined everything up perfectly, makes it run faster, smoother, it's more efficient. Over here, it doesn't have everything lined up. Okay, it's pointing this way. You can see how it's kind of looks like it's not very straight there. We're not going to get as smooth or as efficient production there, and so that's why it produces less vertebrae. And that's what adds to different organisms as well. Okay, and they have the same exact gene, just different enhancers. And enhancers are a site that allows enhancer proteins to bind to it, just like the promoter site. And so that's why they say a lot of the human genome isn't even coded for proteins. It just helps in the aids in the production of proteins, and that's what adds those 80,000 proteins compared to our 20 to 25,000 genes. All right, now to translation. So what is translation? Well, translation occurs outside of the nucleus. Outside nucleus. Now it either occurs in the cytoplasm of free ribosomes. Okay, it's either going to occur on the free ribosomes in the cytoplasm, or it's going to occur on the rough ER, depending on what type of protein it is. Now, translation is the process of going from mRNA to protein. It's that final step in going from genotype to phenotype. We're going from an mRNA molecule, that message that the DNA sent out, to the protein that will ultimately function in our body, make us who we are. So let's take a look at the steps of translation. Again, there's 3 steps. There's a little more involved, though, than transcription. Here we can see, before we get going on the steps, is there's 2 parts to the ribosome. You have the small unit down here and the large unit up here. Okay? The large unit has 3 sites, and I call them APE. There's one here called A, one here called P, and one here called E. So it's either EPA or APE, however you want to call it. Even though it's backwards, I call it APE. Helps me remember that there's 3 sites, APE. I'm not concerned that you need to know, I'm not going to worry that you know APE, the A, P, and E sites. I just want you to know where these tRNA come in. So a lot of times remembering APE will help you figure out where the tRNA come in order to build the proteins. Okay, and that'll make more sense in a minute. So let's look at the first step of translation. Again, these terms are really easy. First step is initiation. So what happens in initiation? Well, what happens in initiation is the mRNA molecule binds to the small unit of the ribosome. So this mRNA molecule right here comes in and binds to the ribosome. As that happens, our initiator tRNA, so our first tRNA, which is this guy right here, Okay, we'll put a 1 on them in red so we can see it. 1 comes in and binds in the P site, okay, and it binds to what we call the start codon. Okay, it binds to the start codon. The start codon for us is always going to be AUG. Okay, and I'll redo that here. Always going to be AUG. Okay, that's always going to be the start codon in mRNA. There may have been a bunch of stuff here, but a lot of times that's what we call cap and tails. They're actually little protectors. Okay, so we always want to find the first AUG on the mRNA strand, and that's where we'll start coding. Okay, so the first tRNA molecule comes in, binds in the P site here, and binds with its anticodon to the start codon AUG. Okay, remember anticodon is the ones on the tRNA that help stick together. In this case, it would be UAC. That would be the anticodon, UAC. All right. At this point, the large part of the ribosome also comes together, so it becomes one functional unit in initiation. Everything is set up to produce the protein. Okay, that's what happens in initiation. Everything has to come together and set up to produce the protein. Now this step right here and into these steps right here is part of the second step of transcription, which is called elongation. And what happens in elongation is, and I'm going to just clear all this ink actually and start over. Elongation. Again, this is the second step. What happens in elongation is another tRNA comes into the A site. Remember, that's the first one here, the A site. And it binds to the next codon, the next set of 3 bases. Alright, and that's all based on its anticodon. Now notice these tRNAs are also bringing in amino acids here. Once the tRNA comes in and binds to the mRNA, these amino acids will link together. But they'll stay on this second tRNA. Once they link together, the tRNA molecules shift down with the mRNA. So the mRNA moves, the tRNA move with them. Okay, everything shifts that way, except for the ribosome. And what that does is it opens up another site, and it opens up 3 new bases for another tRNA to come in and bind. Now the original tRNA is going to leave without the amino acid. A new one comes in, and then these 3 amino acids will bond together, and then everything will shift down again, and it would keep happening until we coded the mRNA strand and we brought in enough amino acids. Now, how do we know which amino acid to bring in? That's based off the codons. Those 3 bases in a row is what tells us which amino acid to bring in. Okay? And that's RNA elongation. Now the final step is termination. And so what happens is, if I clear this page here, in termination we reach a stop. Let's say this is the last 3 codons here, and it's a stop codon. Stop codons are segments of the mRNA that tell the ribosome and the tRNA to quit coding. That's the protein. You've had enough amino acids. You've made the protein. You're done. And there's 3, and you don't need to memorize these, but here's 3 stop codons. So any of these segments would tell them to stop. Okay. And again, this is step 3, so termination. And in that case, we would say, okay, well, we got our 3 amino acids, our protein's formed. Now, we would need 150 or more amino acids to make a protein, but for this simple case, that would be translation. Initiation, elongation, termination. Here's a more up-close look at how the tRNA attaches with its anticodon to the codon. Again, codon is 3 bases together. We're always going to start coding. We're always going to start coding with the start codon, which is AUG, and then we'll stop with a stop codon. And that's all found on the mRNA molecule. Now, how does these codons code for amino acid. Well, what we figured out is that there's certain segments that code for certain amino acids, and so we're able to look at this nice, beautiful amino acid chart to figure out what amino acids coded for by certain segments of bases. So what you would do is you would find the first letter of the base. So let's say we're looking at the codon AUG, right, our start codon. And we want to know what amino acid's coming in for the start codon. Well, we find A here, then we find U, then we find the segment that's AUG, and there we go. So the amino acid is this MET, which actually stands for methionine. You don't have to memorize this chart. You'll always have it for an exam. You'll always be able to look it up, and you don't ever have to write out the full name. Okay? But you need to know that's what a codon codes for, and it's the codon on the mRNA that codes for these. The tRNA just brings the amino acid in. Again, here's another picture. Here's a little better picture of how we show RNA elongation. So you can see how we're building this long strand of amino acids. They're just going to keep coming in and keep building that amino acid chain. And the other tRNA are just going to keep leaving. Okay, and that's part of the RNA elongation. Or sorry, just elongation, not RNA elongation, just elongation. And then here would be a fully functioning protein here. This shows you the sequence of amino acids. It also has the finished functional groups on the end that the rough ER or the Golgi would put on in order to finalize that protein. Okay, so that's ultimately what we're trying to make right there, is a protein through transcription and translation. All right, there ends the lecture on transcription translation. Make sure you go over this, take good notes, and also watch the lecture on DNA replication as well. Take care and good luck on the exam.